Lecture 23, covering Chapter 18, the evolution of invertebrate diversity. So we've moved on. We talked about our prokaryotes. We did an introduction to eukaryotes in the form of protists and plants and fungi. And now we are moving into animals. So we're going to be talking specifically about invertebrates today. And then in the next lecture, we will get into vertebrates. So to give you an introduction, and it goes straight into insects, because when we talk about invertebrates, we're usually talking about insects. Insects encompass a wide variety of shapes, sizes, habitats, diets, mating habits, other characteristics. With more than 1 million species of insects that we know of, it comprises nearly 3/4 of all the animal species. There are more than just insects that are invertebrates, but quite often it's going to be an insect because there are so many insects. I have a song about it that I might sing for you later. So we're going to talk briefly at the beginning of this lesson about animal evolution and diversity in general. And then for the rest of this lecture, we're going to focus on invertebrate diversity. And then in the next lecture, we're going to talk specifically about vertebrate diversity. And again, the difference between invertebrates and vertebrates: invertebrates do not have a backbone, vertebrates do. And let's get into animal evolution and diversity first. Just a quick little primer before we jump into invertebrates. So what is an animal? Right? There are lots of different types of animals. Think about the fact that we're an animal, and so is that fly that is currently terrorizing my poor dog. So animals are eukaryotes. They're multicellular heterotrophs. So they're composed of many cells, and they're heterotrophs. So they are consumers. They have to consume their organic material. And they have cells that lack cell walls. Remember, our plants had cell walls, our animals do not. So animals use ingestion. They eat food in order to get energy. And animal life cycles and embryonic development also distinguish them from other groups of organisms. So if you take a look, this is an example of a sea star. So mammals are going to be a little bit different, but we're talking about invertebrates today. So I'll give you an example of an invertebrate. It's similar to what you'll see in mammals though. So we have an adult, in this case, an adult sea star. And again, our orange is haploid, our blue is diploid. So we have a diploid adult sea star that is going to do meiosis to produce, in this case, an egg. Then you are going to get fertilization of the egg by the sperm, creating a diploid zygote, that fertilized egg. So we're back to diploid. It's then going to grow, become multicellular. So there's an 8-cell stage in this case. Then we go into a blastula. So it keeps dividing, right? We start off with just a zygote. It divides, becomes the 8-cell stage. That's going to further divide into new cells, creating a blastula. That's going to further divide to make an early gastrula. That's going to further divide, and that's going to make the later gastrula. And then, since we're talking about our sea stars here, it is going to form into a larva, which that larva is then going to undergo metamorphosis to become an adult. So you can think of the larva like the juvenile form. It undergoes metamorphosis to become an adult, and then the cycle starts again. So a little bit different than you'll see in mammals, where you'll get, you'll still get fertilization and you'll get the division of cells, but eventually we end up with, something like a baby, which then just grows into an adult. A lot of our invertebrates are going to start off as larvae as their juvenile form, and then go through some kind of metamorphosis before they become an adult. So animal diversification began more than 500 million years ago. The oldest animal fossils are from the late Ediacaran period, about 560 million years ago. They're actually impressions of soft-bodied animals that varied in shape and length. Animal diversification accelerated rapidly during the Cambrian explosion from 535 to 525 million years ago. So there's a lot of words that I'm reading on the screen. I want to call that out to you, the Cambrian explosion. So from about 535 to 525 million years ago, there was this period known as the Cambrian explosion. During the Cambrian period, we call it an explosion because we went from a few animals to a whole bunch of animals fairly quickly. So 10 million years doesn't, it seems like a pretty long amount of time, but considering the Earth is, 7 billion years old, or 4 billion years old, there's That's very quick in evolutionary terms. So Cambrian explosion, 10 million years, all of these species actually happened fairly quickly. So the Cambrian explosion is a really big deal. I want to call that out to you because it was like all of a sudden we got all of these new animal species. In contrast to the uniformly soft-bodied Ediacaran animals, many of the Cambrian animals had hard bodies like shells and spikes. So we're getting some new types of animals in here as well. So of the 35 or so animal phyla that we know of, systematists disagree on the precise number, but there's about 35 or so animal phyla. Remember, kingdom, phylum. Only one of those phylum includes vertebrates, the animals with a backbone. All the other 34-ish are invertebrates. So there's a lot more invertebrates. About 96% of animals are invertebrates, the animals that lack a backbone. So many more invertebrates than there are vertebrates. So animals can be classified, they can be characterized by basic features of their body plan. So some of the different ways that you can characterize animals can be by their symmetry, by the presence of true tissues, by the number of embryonic layers, the presence of a body cavity, and details of their embryonic development. So a lot of what we're going to do over the rest of the lecture is we're going to be looking at different phyla of invertebrates, and we're going to be classifying them. So we're going to be going through some of these different body plan characteristics in order to understand why they're in the group that they are in. So there are two types of symmetry that I want you to be familiar with. There is radial symmetry and there is bilateral symmetry. So an example of radial symmetry is a sea anemone. Radial symmetry means it's kind of a circle, and no matter where you cut it, the two halves would be the same. In bilateral symmetry, on the other hand, you would have to cut it very precisely down the middle in order for the two halves to be the same. So radial symmetry, and then a circle. Bilateral symmetry, you'd have to cut it right down the middle, something like a lobster. Body plans and molecular comparisons of animals can be used to build phylogenetic trees. So biologists make hypotheses about the phylogeny of animals, the grouping of animals, using evidence from the body plans that we just talked about, the fossil record, and molecular data, chiefly DNA sequences. All right, so that is our brief primer on animals. We're going to jump into invertebrate diversity. Again, we're going to be looking at the major phyla of invertebrates. So you're probably familiar when we're talking about animals, or when we're talking about mammals specifically, we are usually talking on the genus and species level. We talk a lot about the species of animal, that's usually mammals, because there aren't as many of them. When we're talking about invertebrates, we're usually way up at the phylum level because there are so many of them that's the level that is manageable. So we're going to be talking about different phyla of invertebrates today and what characterizes each of them. So here is your first phyla. So this is phylum Porifera. Phylum Porifera, and porifera are the sponges. So sponges are animals, they're synthetic sponges too. So the one you have on your kitchen counter might not be a real one, but some of them are. So phylum Porifera, these are our sponges. Sponges are relatively old organisms, old in evolutionary terms. And so they're relatively simple and have porous bodies. They're very simple animals. They do not have true tissues. They usually don't even have any body symmetry. They don't have the radial symmetry. They don't have the bilateral symmetry. They're just kind of blobs. And they're usually marine, although some of them are found in freshwater as well. They also have what's called flagellated choanocytes. Flagella are those little whip-like tails that help organisms move around. So they have flagellated choanocytes, which filter food from water passing through pores in their body. So phylum Porifera, those are our sponges. You can see a picture of a sponge in the background. There's lots of different kinds. I encourage you to Google sponge and look at some pictures of them. But sponges, relatively simple, no true tissues, lack body symmetry, usually marine. So there's your little checklist for sponges. So, here are some different sponges. They look a little bit different. They're fun when you scuba dive. Okay, moving on. It's going to be a quick tour. It's going to be a quick tour, my friends. We're moving into phylum Cnidaria. These are known as cnidarians. They're radial animals with tentacles and stinging cells. So Cnidaria means stinging cell. And so these are organisms like our jellyfish and our sea anemones. So notice that we refer to them as jellyfish, although they're not actually fish. They are invertebrates. They are cnidarians. They're characterized by radial symmetry. They have 2 true tissue layers. They also have a gastrovascular cavity. Gastrovascular means where they digest their food. So if you're thinking about sea anemones, they have stinging cells on their tentacles. As the water flows by, they will actually use those stinging cells to catch their prey. The mouth is in the middle, so they will pull all of that in and they will get the food off. They digest it in their gastrovascular cavity, and then they spit out the waste through the same hole that it entered. So one hole there doing both things. There's 2 body forms to our cnidarians. There's the polyp and the medusa. So the polyp form is this one, where the base is stuck on a rock and the tentacles stick up. And so those are our sea anemones, right? You can think of it like this. It's easier with one hand, right? So this is our polyp form. The other form is medusa. Just flip that over. It's like a jellyfish. So that's where the base is at the top and the tentacles reach down. And they actually kind of switch. So like jellyfish will start off their life, their larval form, as a polyp, and then they detach and become a medusa form. So that's the jellyfish. That's also why in Spanish the word for jellyfish is medusa, right? Medusa form. But those are cnidarians. Cnidaria means stinging cells because they do have stinging cells. A fun fact for you, not that I encourage you to go poke animals at the tide pools, but if you ever get a chance to go to an aquarium where they have a touch tank and you're able to touch a sea anemone, you'll notice it feels kind of sticky. That's actually it stinging you, but the skin on our fingers is so thick that their stinging cells can't make it all the way through. So it just kind of feels sticky to us. But those are our cnidarians, stinging cells, jellyfish, and sea anemones are 2 examples. Corals, by the way, corals are also in this group. So corals, like that form coral reefs, are animals. They're colonial animals in this group. So they are related to sea anemones and jellyfish. So we have a hydra, we have a sea anemone, both examples of cnidarians. Then we're going to move into our next phylum. Are you ready? We're going quick through this one. Now we're into phylum Platyhelminthes. Platyhelminthes are our flatworms. They're the simplest bilateral animals. So we had porifera, our sponges, which did not have any symmetry. We had our cnidarians, the stinging celled organisms that had radial symmetry. Now we're into Platyhelminthes, our flatworms, which have bilateral symmetry, so you can cut them down the middle. They're very simple organisms. They do not have a body cavity. There's a flatworm called a planarian, which has a gastrovascular cavity and a simple nervous system. Flatworms live in marine, freshwater, and damp terrestrial habitats, but notice they're all tied to water. And there's also flukes and tapeworms in this group, which are parasitic flatworms with very complex life cycles. So you can see an example of a flatworm here. Look at its mouth. It's down here. Has eye cups, so they're able to sense light and dark. Nervous tissue clusters up here. There's a gastrovascular cavity helping to digest food. Some nerve cords running along here. And here's our bilateral symmetry. The poor little flatworm, look at its eyes. It's like, oh, or it's, I guess it's eye cups. All right, next phylum. You ready for it? Phylum Nematoda. These are our nematodes, also known as roundworms. Phylum Nematoda has a body cavity and a complete digestive tract. So we've looked at organisms before that have an incomplete digestive tract. The big difference between the two is in an incomplete digestive tract, the same hole where food enters is the hole where waste leaves. We're talking about a complete digestive tract that is mouth as one hole, anus, the waste expulsion hole, as a separate hole. And I challenge you to think about the advantages of a complete digestive system. It's probably exactly what you're thinking. So nematodes in our phylum Nematoda have bilateral symmetry. They have 3 tissue layers, a non-living protective cuticle that's shed periodically, and a complete digestive tract with a mouth and an anus. Yay, it evolved. Some nematodes are free-living decomposers, while others are plant or animal parasites. All right, pause. So you realize we're moving on to the next phylum. Now we're in phylum Mollusca. These are known as the mollusks, sometimes spelled with a K, both are correct. And mollusks are our snails. So mollusks are incredibly diverse, and there's lots of different variations on a common body plan. So some of the common characteristics of our mollusks are that they have a muscular foot, which functions in locomotion. So think of a snail, that's that large part that's going to be attached to the ground that is helping them move. That is the foot. There's a visceral mass, which contains most of the internal organs. So that's kind of the other part of its body. There's a mantle, which is where a shell may or may not be secreted, and the shell, there may or may not be a shell that encloses the visceral mass, and they also have a circulatory system. Many mollusks feed with a rasping radula, which is used to scrape up food. So you're not going to see a bunch of teeth on these guys. Instead, they have a rasping radula, which helps just scrape up food. And there are many different types of mollusks. There are slugs, which do not have a shell. There are some that have just one shell, think of snails, and then those are bivalves, which have two shells. So think of clams and oysters and things like that. There are some on land, like our snails and our slugs. There's also a lot of mollusks in the ocean. So if you're thinking about, there's lots of sea slugs. There's also sea snails. We have our turban snails, our limpets. There's clams and oysters and mussels and all kinds of mollusks that live in the ocean. Sea hares is a type of slug. If you've never heard of a sea hare, look it up. They live in our tide pools and they're super cool. But snails, you're probably familiar with them. That is in phylum Mollusca. All right, moving on to phylum Annelida. Annelids are segmented worms. So segmentation is the subdivision of the body along its length into a series of repeated parts, so different parts of the worm. They have a nervous system. They also have a closed circulatory system in which blood remains enclosed in vessels throughout the body. So circulatory system, we have a closed circulatory system as well. Many invertebrates, such as mollusks and arthropods, have an open circulatory system in which blood is pumped through vessels and into open body cavities. Not the annelids, they have a closed circulatory system. It's going to stay in the circulatory system. And there's two major clades of annelids. There's the Arantians, which are mostly mobile marine worms, and there's the sedentarians, the less mobile worms. Look at that, sedentarians, because they're sedentary. Get it? Get it? These are earthworms, leeches, and many tube-dwelling worms. So annelids are segmented worms. Here's some pictures just to show you some diversity. You can see the segments here. See the segments? That's why they're called segmented worms. Now we're into arthropods, and we're going to spend a little bit more time on arthropods because there are just a lot more arthropods. So this is phylum Arthropoda. And there's a song, phylum Arthropoda's got a quintillion wriggly things in it. It starts off with, I swallowed a bug just the other day. What kind it was, I just can't say. Anyway, now you're gonna remember phylum Arthropoda's got a quintillion wriggly things in it. These are our insects, bugs, as well as things like crayfish, lobsters, crabs, barnacles, spiders, ticks, and insects are all part of phylum Arthropoda. So we have marine creatures like crayfish, lobsters, crabs, and barnacles. Oh, this is a college class. I love teaching college because I can tell you my favorite fact about the animal kingdom, barnacles. If you've never seen barnacles, Google it right now. This is going to really interest you. Barnacles look like little volcanoes, but they're animals. They are arthropods related to lobsters and crabs. Look like little volcanoes. They're about yay big, white, on rocks in the tide pools. They don't look like much, but they are animals. And barnacles have the largest penis to body size ratio in the animal kingdom. You're welcome. Now let's move on to insects. So arthropod diversity and success are due to their segmentation. They have a hard exoskeleton. So that's why if you go to the tide pools and you step on barnacles, that doesn't really hurt them. Think of lobsters and crabs. It's not a shell, it's an exoskeleton. They also have jointed appendages. So that arthropoda actually means, we've heard the word poda before for feet, right? So arthropoda means jointed appendages, which is why it's named phylum Arthropoda. And as an arthropod grows, it must periodically shed its old exoskeleton and secrete a larger one. This is a process known as molting. So if you've ever been to the beach, you've probably seen, it looks like there's a lot of dead lobsters on this beach, but it's not actually dead lobsters, it's the molt. So the exoskeleton of lobsters and crabs and other arthropods, it doesn't grow with it. They have to molt it. They shed it, and then they grow a new one that's larger. So that's the process known as molting, also known as ecdysis. So you can take a look at a lobster here. It's got these little swimming appendages in the back, and it has a segmented body again. So it has the abdomen, it has a thorax, it has its head, has our antennae up here, which are used for sensory reception. It's got these little walking legs, mouth parts, and then pinchers for defense. This is the horseshoe crab. They're super cool because they have blue blood. But they're another marine creature. They're about yay big. They're real pokey if you try to pick them up. They're also real feisty if you try to pick them up. I've worked in a lot of aquariums and other places where we were caring for these in captivity. I don't just go walking down the beach like picking up and throwing horseshoe crabs, I promise. But there's a lot of medicinal uses for these. So a lot of them are harvested out of the ocean in order to make medicine. Here are some more arthropods. So we have scorpions are arthropods, spiders, dust mites are arthropods. There was some statistic that I heard, something like if you were to go and put a bug net over a tree and like collect all the bugs in that one tree, that you would find more species of bugs than like all of the species of mammals that exist in the world, something like that. There's so many arthropods, there's so many insects. And remember that all insects are arthropods, but not all arthropods are insects. But insects are the most successful group of animals, and nearly 75% of all identified animal species are insects. There's thought to be as many as 30 million insect species. We don't know because there are just so many of them. They live in really weird places and they're hard to spot. They're also hard to tell apart sometimes. So we think there's about 30 million insect species, and we think that mainly because we've done some math on it, not that we've identified that many. The body of an insect typically includes a head, a thorax, an abdomen, 3 sets of legs, and wings in most of them, but not all of them. Insect life cycles often include metamorphosis, during which the animal takes on different body forms. It develops from larva to adult. Think of a butterfly. You're familiar with metamorphosis, from a caterpillar to a butterfly. More than 80% of insect species undergo what's called complete metamorphosis, in which a free-living larva, that juvenile form, transforms from a pupa into an adult. So completely different. There's other insect species, though, that undergo incomplete metamorphosis, in which the transition from larva to adult is achieved through multiple molts, but without forming a pupa. So complete metamorphosis is kind of like, boom, it's now an adult, right? Like think of like a caterpillar, where it's going to enclose itself in a cocoon and form a pupa, but then it emerges as a full butterfly, right? That is complete metamorphosis. There's others, though, that undergo incomplete metamorphosis, where there's just going to be little steps in between. So they still transition from larva to adult, but that's done through multiple molts. So they're going to molt one, and they're going to be a transitionary form. They'll molt that, become another transitionary form, until eventually they become an adult through multiple molts, and they don't form a pupa. Nearly 75% of all identified animal species are insects. I think that's like the 3rd time that I put this in here. There may be as many as 30 million. Ooh, that might have been a copy and pasting error, my friends. I'm sorry. Oh yeah, that's copy and pasting error. We're just gonna move on to the next one. So take a look at the insects here. We have our larvae. So here's complete metamorphosis. Larvae, little grub, is then going to form a pupa. And from that, it's going to emerge as an adult. Our little horned beetle here. So that is complete metamorphosis. And insects, again, are the most successful group of animals for several reasons. They have that body segmentation that I talked about earlier. They have the exoskeleton. So a lot of people confuse exoskeleton for shell. They're not the same thing, although they are used for protection. So they have an exoskeleton, they have those jointed appendages. A lot of them can fly. They have a waterproof cuticle, so a lot of them are very good in the water. And they have complex life cycles with short generations and large numbers of offspring. So insects don't live very long, but they sure have a lot of babies. They also have really, really great camouflage. I mean, think of the insects that you've seen, really amazing camouflage. And there's so many insects that have evolved all over the place due to these. So take a look at this. I think this one's actually a moth, but you can see one of the common traits are, one of the common adaptations are these eye spots. So they're not actually eyes, they're just eye spots. It's just coloration that looks like eyes. But if you were a predator and not as intelligent as each of you are in this class, you might mistake these for eyes and think it's a big old animal looking at you. You can also probably guess where this moth lives. It probably lives on tree trunks because it looks kind of like a tree trunk, right? So just some incredible, incredible camouflage within insects. So there were arthropods. We still have a few more though. We still have a few more phyla. So that was phylum Arthropoda. Now we're moving into phylum Echinodermata, which might be my favorite invertebrate phyla. Yes, I have a favorite, and it's Echinodermata. So these are our echinoderms. Echinodermata means spiny skin. Echino here means spiny, derm means skin. You think of the dermatologist for skin. So Echinodermata, spiny skin. So these are organisms like our sea urchins, our sea stars, our sea cucumbers. They have an endoskeleton. So our arthropods had an exoskeleton, a skeleton on the outside, not exactly a skeleton, but on the outside. Our echinoderms have an endoskeleton. So theirs is going to be on the inside. So if you've ever seen a dead sea urchin, those are those spiky purple balls that you find underwater. We have some at La Jolla Shores. There's actually a big population of sea urchins at La Jolla Shores. So go for a swim, take some goggles down there and check it out. But if you've ever seen a dead sea urchin, that endoskeleton is called a test, just like the test that you take in school. But they're white and they're hard, and that's what's left behind. That's that endoskeleton. What am I talking about? Sand dollars. Sand dollars are another echinoderm. They also have a test. When they die, when they're living, they're purple and they stand upright. When they're dead, they're white and they flip over. And sometimes you'll find they look like cookies with a star on them. Those are the sand dollar tests that will wash up on shore sometimes. There's also some of those at La Jolla Shores. So our sea urchins you can find in the tide pools and some of them down in the kelp forest. Our sand dollars is a huge population out by the submarine canyon off of La Jolla Shores as well. So they're a really diverse group: sea stars, sand dollars, sea urchins. They're usually slow moving or they're sessile, which means that they don't move around. They're all marine. They have radial symmetry. They actually have specifically what's called penta-radial symmetry. So penta means 5. That's really obvious in our sea stars, where they have the 5 arms, right? I'm trying to get you the 5 arms. And while it's not as obvious in the sea urchins and the sand dollars, if you look at their tests, that endoskeleton they leave behind once they die, you can actually see. So on the sand dollar, it has a star with 5 points. You can see the penta-radial symmetry. So that means they have radial symmetry. It's actually divided into 5 pieces. Echinoderms, again, have an endoskeleton of hard calcium-containing plates under a thin skin. They have a water vascular system based on a network of water-filled canals that branch into extensions called tube feet. So tube feet, if you've ever looked at the bottom of a sea star, they have these little suction cups at the bottom they use to move around. Those are the tube feet. So the water vascular system actually connects through canals to those tube feet. And they're able to kind of inject water into those little tube feet, and that's how they can walk around. They also have the ability to regenerate lost arms. So if a sea star loses an arm, it can grow a new one. So if you take a look, right, here's our sea star. You can actually see the pentaradial symmetry, the 1, 2, 3, 4, 5 different arms fairly easily in a sea star. They have these little spines. Sometimes they're hard, sometimes they're soft. Look up the chocolate chip sea star if you get a chance. It's real cute. It has some soft spines. Some of ours, like the ochre sea star here in our tide pools, have harder little spines. Here are the tube feet down at the bottom. Notice that some parts of them stick out. That's how they move around, connected to the vascular system by these canals. They have a stomach inside, and here's the anus. And here's a fun fact about sea stars. So sea stars love eating mollusks. Think mollusks were the snails. So you can imagine though, the stomach of a sea star is on the bottom. So if you flip a sea star over, you'll see all the little tube feet. And there's a hole in the middle. That's its mouth. You'll notice the mouth though is really small. And so they love eating things like mussels. So how do they get the mussel inside? They don't. Sea stars, in order to eat, will actually spit up their stomach. Like they throw up their stomach outside of their body, digest the food, and then pull their stomach back in. It's so interesting. They'll actually attach to the mussel using their tube feet. They'll pry it open, throw up their stomach, digest the yumminess, pull their stomach back inside, and then go on their way. They're really cool. See, look at this. Oh, and then sea cucumbers. Look up sea cucumber. They look like they're usually orangish or red, and we find them in tide pools, and we find them down in the kelp forest as well. They're usually about yay big. They can be a little bit bigger. Some people think they're delicacies. Some people eat them. But one of the neat things that they do for protection is something called evisceration. So evisceration means that if they are threatened by a predator, rather than letting the predator eat their whole body, they will actually expel some of their guts. They like poop out their guts. And then while the predator is busy eating their guts, they hightail it out of there. And since they are echinoderms, they're able to regenerate. So they regrow their guts. Evisceration. We used to play a game with the kids called Evisceration, where we'd be in kayaks and throw balls at each other, simulating that. Oh, teacher. Oh, teacher. Okay. So those are the echinoderms. Take a breath. Moving into phylum Chordata. So phylum Chordata is our own phylum. It's distinguished by 4 features. The dorsal hollow nerve cord. So that's where we get chordata from. So now we're in the chordates. These are going to have a spinal cord. Not all are vertebrates though. Vertebrate is an animal with a backbone. So we're moving into spinal cords, but not necessarily into backbones yet. So backbones are going to, those vertebrae are going to protect the spinal cord. So we're at spinal cord now, but not necessarily vertebrae. We'll move into vertebrae in the next lecture. But our chordates, phylum Chordata, have a dorsal hollow nerve cord. They have flexible, supportive, longitudinal rod called the notochord. They have pharyngeal slits and a muscular post-anal tail. Ooh. So let's look at a few examples. So we have tunicates and lanceolets, which are what we are going to talk about today because they are chordates that are invertebrates. And then we are going to be moving into our vertebrates in the next lecture. So we're going to briefly cover our tunicates and our lanceolets. They don't have a backbone. So remember, they have that spinal cord. So they are chordates, but they are invertebrates still. So they don't have a backbone protecting that spinal cord. And they use their pharyngeal slits for suspension feeding. And so they have these little slits that they're able to pull nutrients out of usually the water. Adult tunicates are stationary and attached, while tunicate larvae are more like tadpoles. They're not actually tadpoles, but they're tadpole-like organisms. And then lanceolets are small blade-like chordates that live in marine sands. So they look kind of like worms, but they're not. Tunicates and lanceolets are in phylum Chordata, so completely different phylum than our worms, which are in a couple different other phyla. So you can take a look here. We have our post-anal tail. Post-anal means after the anus. We have our anus here. Look at that. The tail is after the anus. We have segmented muscles. We have this digestive tract going down the middle. We have our pharynx, our mouth, our head. Here are the pharyngeal slits with the pharynx. And you can see the notochord and the dorsal hollow nerve cord. Here's another picture for you. And I want to take a brief moment to touch on the importance of invertebrate diversity. It's a valuable but threatened resource. Invertebrates play critical roles in natural ecosystems and provide valuable services to humans. So a lot of my job as a conservationist is saying, if you don't care about the animals in their own right, care about what they do for you. And invertebrates do a lot for you. So A lot of invertebrates like freshwater mussels filter and improve water quality in natural ecosystems, and they reduce the cost of water treatment for human uses. So instead of having to put a water treatment plant, which is expensive, if we have native freshwater mussels, they're able to filter and improve water quality on their own. So they save us a ton of money and effort. There's reef-dwelling cone snails that produce a powerful painkiller in their venom. There's flowering plants that are pollinated by animals, chiefly insects, and flowering plants usually lead to fruit. Some of that is the fruit that we eat. So they provide us with a lot of food. Also, think about how many invertebrates you eat. I've been mentioning things like clams and oysters and snails. A lot of that is food. Sea urchins, which is uni, I think. A lot of especially marine organisms are used as food. Arthropods, insects, food, right? A lot of invertebrates are food for humans. And so that is why it is so important to help protect species like this, because they do a lot of stuff for us. All right, so you should now be able to describe the defining characteristics of animals, describe the general animal life cycle and the basic animal body plan, describe that Cambrian explosion. What was it? Why does it matter that we have a lot of different animal diversity? Characterize, we talked about 9 animal phyla today. So we talked about 9 phyla of invertebrates, be able to list them and give the general characteristics of each. What is segmentation? Why is it important? Where are the phyla that it occurs? You know, we talked about arthropods. Tell me why arthropods are important and some of the different characteristics of them. Describe the common characteristics of insects and explain why invertebrate diversity is a valuable resource to humans. So that is the end of this lecture. I will see you for vertebrates in the next lecture.